So hopefully we can see that 1 turn of the cycle is not enough. 3 turns of the cycle is what's needed to produce that one stable molecule of TP that can leave. The rest get regenerated to RUBP. So to highlight this, there's a slightly sort of more stylized sort of diagram there, which goes through the whole thing. So RUBP is a 5-carbon sugar, and it joins with the CO2 to form the 2 molecules of GP, this glycerate phosphate. That's a fixing process, carbon fixing, and it uses that long-winded enzyme there, which luckily we can abbreviate to Rubisco. Now, interesting enough, as a little side point, Rubisco is incredibly useless as an enzyme. It's very ineffective. I like to think of it as the Segway of enzymes. It's only a little bit better than walking. You know, if I said, let's go to Scotland and we decided to take segways, you'd be like, why don't we fly? That's pretty much what the enzyme's like. It's pretty useless, but it does pretty well for what it does. Obviously, nature has decided that's good enough for purpose, so it's continued. And it compromises 50% of the mass of the chloroplast. It is the most abundant protein in nature. Really good pub quiz question there. And it's used to synthesize the GP from the RUBP using CO2. And it uses the ribosomes within the chloroplast itself, and it uses the chloroplast DNA. It doesn't have to annoy the nucleus, for want of a better term, for the genetic material. It just goes, well, I've got it in situ, I'm going to make more of it. From there, the GP is reduced using the NADPH, or reduced NADP, and also the energy from ATP. ATP provides energy, NADPH, or reduced NADP, provides reducing power. And they then are spent effectively or used, and they go back to the thylakoid membrane to be recharged in a light-dependent reaction. From here, we've got a branching point. So TP can go in one of 2 directions. It can either continue around the cycle to make more RUBP, so the carbons can be regenerated, so into the RUBP, which is quite complex, but remember, it does take a couple of units of ATP to do this. It's not just an instantaneous thing. TP to convert back to RUBP takes energy. And RUBP itself is a very complex molecule. It's pretty involved, and it is a carrier molecule. It can pick up CO2 and convert it. That's the whole idea of it. So this molecule can be broken down and rebuilt again. So it's just a carrier molecule to help convert the CO2 into ultimately glucose. So that's one section of where the TP can go. The other one is it can leave the cycle. For every 3 turns, 1 spare TP is manufactured, which could then leave the cycle, and 2 TPs together can make glucose. So we've got this 2 molecules joined together, glucose, which can be stored as starch or can make cellulose, amino acids, lipids, etc. It can be used for any of the plant's needs. So when you look at the light dependent and light independent side by side, it becomes incredibly complex. We've got the ATP being manufactured, which is then going to go and convert the GP to TP. We've got the reduced NADP, which is being synthesized, which is going to help with the conversion of GP to TP. Also, that ATP can be used to regenerate RUBP. So there's an awful lot going on. It's best if you sort of view them separately and then try and link them together afterwards. So this all takes place in the stroma. It's the fluid-filled space within the chloroplast itself, and it's where all the enzymes that are involved in the reduction of CO2 are found, and also it's where the products for the light-dependent reaction can go and power this cycle. So the light-independent reaction is not dependent on light directly, but it will grind to a halt if there's no light eventually, because these products will run out. Also, as I mentioned before, there is DNA and ribosomes within the chloroplast, which are going to help manufacture all the essential proteins for this. And the final thing to finish off on is Melvin Calvin's experiment, the Lollipop experiment. You can see clearly there why it was called the Lollipop experiment. And it's actually a remarkably simple experiment, but it was pure genius how it's worked out. The screens on either side there are to protect the algae, which is in the green lollipop, from excess heat. And it was a remarkably well thought out practical. So what we've got here is a very simplified version and a very bad diagram of Melvin Calvin's Lollipop experiment. There's a few things we need to consider. The first thing is the green section here is the algae within the nutrient medium. So it's got all the things to keep it alive. We've also got to remember that air and CO2 will be allowed to go in so it can photosynthesize. Also, if you remember the picture we saw last, there was light on either side. So it's definitely got enough light to photosynthesize. What we want to do is we want to introduce to it radioactive CO2. So you plunge the plunger in on the syringe and radioactive CO2 is introduced to this. A timer is then started, and every 5 seconds, the tap at the bottom, the little T-like thing here, is turned and algae drops from the lollipop into the hot methanol here, which will kill the algae, instantaneously freezing it in its metabolic process. What Melvin found was that at 0 seconds, the only thing that was radioactive was the CO2, which is kind of logical. As soon as you plunge the plunger in, CO2 is going to be the only thing that was in the hot methanol here, which was radioactive. Then after 5 seconds, he found that GP was, remember he was freezing it each time. So every 5 seconds, he was taking another sample and seeing what was radioactive within it. After 10 seconds, he found that the GP and the TP were radioactive. After 15 seconds, he found GP, TP, and glucose were radioactive. After 20 seconds, he found that not only the GP, the TP, and the glucose, but also the RUBP. So if we think about it, if we go back to our cycle, CO2 gets combined with non-radioactive RUBP at this moment and becomes radioactive GP. That GP then gets converted to radioactive TP. That TP can then get converted into glucose, and most of the TP can also be regenerated back into the RUBP. So he proved the cycle through this very simple but pure genius experiment. Anyway, hope that helps. Thanks a lot. Bye.